There is also evidence that this HIV can enter by endocytic mechanism. My name is Ari Helenius, and I'm from the Institute of Biochemistry at ETH Zurich in Switzerland. My binding to receptors on the cell surface is followed by lateral diffusion of the virus along the plasma membrane, the initiation of signaling events that very often then end with endocytic uptake of the viral particle. Endocytosis can be defined as a process in which fluid, solutes, membrane, and particles are internalized by cells by forming an invagination at the plasma membrane, which then pinches off as a vesicle. And the cargo is then transported in this membrane-bounded vesicle or vacuole inside the cytoplasm. Receptor-mediated endocytosis is a specialized form or specialized forms of endocytosis in which the cargo, the material to be internalized, first binds to receptors on the cell surface, and that then endocytosis occur not just of the cargo itself, but of the whole receptor cargo complex. The receptor binding is very important because it helps to concentrate molecules on the cell surface. And obviously, from what you heard in the first lecture, viruses use different forms of receptor-mediated endocytosis. They bind first, and that they are internalized together with one or the other of their receptors. And the viral particle, for example, HIV in this case, has spike glycoproteins on its surface, which are activated, in this case, by receptor interactions to become fusion active, and the envelope of the HIV then simply fuses with the plasma membrane. This then releases the capsid into the cytosol, and many further events later, the capsid is, and in that case, the DNA made up from this capsid, synthesized from the RNA, will then enter the nucleus. There is also evidence that this HIV can enter by endocytic mechanisms, like the influenza here. This virus is an influenza virus, and the viral spike glycoproteins, the influenza hemagglutinin, allows it to bind to the cell surface, to get endocytosed with clathrin-coated pits and with some other mechanisms. And then in the early endosome or late endosome, where the pH approaches 5.5, the hemagglutinin is conformation changes, and it becomes a membrane fusion factor, and the virus now fuses its membrane with a limiting membrane, releasing the viral genome into the cytosol.